They're incredible animals to watch, right? I mean, they're captivating.
Cephalopods are a curious class of invertebrates known for their large brain-to-body ratios. They include squids, octopus, nautilus, and cuttlefish.
I started studying cephalopods as a grad student in 1989. Been studying them ever since, and I'm still in awe of these animals because every time we dig a little deeper, we come up with new things that they're doing at extraordinary levels. Cephalopods have this infinite degree of movement and flexibility. They can worm their way through tiny holes. There are a lot of different factors that go into supporting those advanced behaviors, right? Everything from molecular level to cellular level to these big brains, etc. How everything is connected. That's what fascinates people.
When Josh Rosenthal was in graduate school, he was studying nerve cells in squid. But he kept running into a problem. When he would sequence the squid's genetic information, it would be different every time.
To be quite honest, as I was discovering this in grad school, my first thoughts was, what a nuisance. I can't get a consistent sequence. I didn't understand what was going on. What I've stumbled across was this molecular level novelty, which is completely unexpected.
Rosenthal had discovered a process in cephalopods called RNA editing. So what is RNA editing? Every cell in an organism's body contains the same genes made up of DNA. This DNA tells us everything we need to develop and interact with our environment. Your cells transcribe the DNA into RNA. That tells your body how to make proteins. You can think about the process like one gene producing one RNA that produces one protein. But genes can produce an RNA that can be edited into different RNAs, which then yield multiple different proteins. That's RNA editing. Cephalopods edit their RNA more than any other creatures on the planet. This all happens in real time while they're alive. This large variety of proteins might be what makes them so behaviorally complex.
Let's think about a cookbook, okay? You have a cookbook that's like our genome, full of lots of recipes. And any one night, you're not going to make all of those recipes, right? So you select, I want to make beef stroganoff tonight. You take that recipe and copy it and bring it down to your kitchen. But let's say you're missing an ingredient and you want to make a change. If you're stuck with the original recipe, you can't make a change. But if you can go and edit that recipe, you can now have the adaptability to change things based on your current environment.
That's what squids are doing when they edit their RNA, generating more than one outcome from a single gene. Humans can edit their RNA too, but while we might have several hundred RNA edits in our brain at any given moment, cephalopods have about 60,000.
The scientists are a funny species, right? They focus often in their own little area, and since you're looking at minutia, it's hard to take a step back at times and see what should be obvious. And this moment came embarrassingly slowly when he thought, wow, if you could harness this capacity in humans, it could be used for medical purposes.
So Josh Rosenthal pitched the idea for an RNA editing biotech company, and in 2019, CoroBio was born.
The goal for us is to harness this technology called RNA editing and develop medicines, not just for patients with rare diseases, but also target large patient populations such as cardiovascular disease, Parkinson's, Alzheimer's.
Most of our medicines now would be to correct the outcomes, the symptoms, but what if you could correct the information itself?
Genetic diseases are diseases of information. They happen because of a mutation in your genetic instructions. It would be like if your recipe for beef stroganoff called for 6 tablespoons of butter, but you wanted to use 3. But just like you could edit the recipe and fix the dish, you might be able to edit your genes and help your disease. There are two ways to do this. If you reprint your recipe book, that would be the equivalent of DNA editing. It's a permanent change. But if you scratch out the ingredient, that's RNA editing. You can always go back and erase it.
When you think of large patient populations with chronic diseases, if you want a permanent change and you want it in all the cells or a set of cells, then DNA editing makes a lot of sense. If you want it to be transient, or if the patient population is very heterogeneous, where you have environmental risk factors, that's where RNA editing would be perfect fit, right?
Koro's idea is to create a therapy that functions like an injectable drug. The drug contains a long-lasting molecule that acts like a magnet and binds to a person's RNA. It then recruits enzymes to make changes to the RNA involved in the disease. Just to be clear, scientists aren't using cephalopod RNA in humans. They're taking inspiration from cephalopods to work with the more limited human version of RNA editing. So far, Koro has tested their drug in monkeys to treat a genetic condition that leads to lung and liver disease. They hope to get to human clinical trials for the same disease within the year. Koro is optimistic that they can have a drug ready for patients in 5 to 6 years.
Once that happens, I think the dynamic around understanding what RNA editing can do, how many diseases it can help resolve, then it becomes more exciting.
When thinking about my early work on cephalopods, it never occurred to me that this was going to evolve into an area that maybe could be useful for a medically related phenomenon. And I think that's the beauty of science, right? You don't know exactly where you're going. You're studying this incredible process, and it leads you from something that cephalopods are doing that's incredible to why cephalopods are using it to say, hey, what about humans?
Am I surprised that it's coming from cephalopods? It has to come from somewhere. The cephalopod is a very interesting creature that can, send your imagination in terms of what is possible.